Welcome, everyone, to another edition of 45 Forward, where our mission is to help you, our listeners from Los Angeles to Long Island, make your second half of life even better than the first. 3 years into the COVID pandemic, some experts believe that we are transitioning to an endemic, headed hopefully toward more normal times. And yet, we're still at a loss. We're confused. Where are we with CDC recommendations? When should we be wearing masks? What's the latest advice about vaccines and boosters, especially if we're among the more vulnerable population, like older people? What's the latest advice about vaccines and boosters? The development of other treatments. In today's episode, Dr. Francis Santiago-Schwarz, a distinguished biomedical scientist and educator, will offer an incisive yet accessible guide to understanding this incredibly complex and developing story of COVID-19. What do we know? What don't we know? And why is it important? Throughout her career, Dr. Santiago-Schwarz has focused on creating programs, courses, and professional partnerships and alliances, and designed to empower people from diverse backgrounds with the knowledge they need to make healthcare decisions for themselves. Today, she'll unravel the immunopathology of COVID, giving us the big picture about variants, providing some answers to frequently asked questions about viruses and how they spread and mutate, and when these mutations make a difference. Dr. Santiago-Schwarz will provide a basic primer on how vaccines, antibodies, and other components in our immune system defend our bodies against these viruses. And looking ahead, she'll offer her perspective on potential new treatments for COVID and what strategies we need to stop the virus worldwide, noting that no one is safe until everyone is safe. So now let's meet today's guest, Dr. Francis Santiago-Schwarz. Francis, welcome to the show.
Thank you very much for having me. I'm excited to be here.
Great. I'm excited to have you. So listen, before we get into, the state of COVID and where are we, tell us a little bit about your background and how you got interested in your journey as a research scientist in immunology from Puerto Rico to New York and LA. So give us a little bit of background.
Well, I was born in Puerto Rico, but my family came to New York when I was only 2. So I lived in Manhattan, first in the Lower East Side and then in Brooklyn. And then of all places, we moved to Charleston, South Carolina.
Ah, wow.
When I was 10. And after being there for a few years, including the desegregation years, that's another story, my family returned to Puerto Rico. And there I entered university at the age of 15. And I went straight into the sciences because I was always a science geek. And it was during one of my science labs at the university, I was only 17, when I fell in love with immunology. And at the time, we were conducting an experiment that you can't conduct anymore because of animal rights guidelines on guinea pigs. And so our professor injected the guinea pig with what I now know to be albumin. And he injected the guinea pig the first time, nothing happened. The second time, the guinea pig wiggled a little bit. The third time, the guinea pig just completely went into convulsions and shock and actually died. And I just looked at the instructor and I asked him, what happened? What was that? He said, that was the immune system. That's an immune system response. He went into shock, anaphylaxis. And I said to myself, wow, the immune system did that. I want to be an immunologist.
Wow.
And that's how it happened.
Wow.
I graduated when I was 19 from college. I came to New York. I worked at NYU, first as a research technician. I got my master's while I was working full-time as a laboratory technician. And then I went on to get my PhD at University of Southern California in Los Angeles. I had met my husband and we moved to LA. And while I was a graduate student, I had my 5 minutes of fame. I discovered a cell type of the immune system.
Wow.
I discovered human dendritic cells, which are the cells that orchestrate the almost the entire adaptive immune response. So
Wow. Wow.
then I went on to study autoimmune diseases and describe dendritic cell development from cord blood progenitors.
Wow. Wow.
And and then I left the lab right before the pandemic hit, and I was making a transition into global health. So I was over 45 and I was reinventing myself. So it's
Huh.
very timely to this program.
Right. Great. That's great. That's great.
And,
Okay.
and that's how that's how my journey got me here.
That's great. Great story. Great story. So let's yeah, I know that, in many ways, I guess, I guess we're going into the third year of this evolving pandemic. And I know in some ways people are just tired and drained and, they're arguing over masks and stuff for it. But I wonder if still we could just take a few moments and say, where do you see us? Where are we sort of in overall in where this is headed, do you think? I mean, I think, we tend to forget. I mean, in the early part of 2020, 2020, wow, it was horrible. And I know that your husband was right at the epicenter in Queens of the pandemic. And so it's not nearly what it is then, thankfully. But where do you see us now in terms of where are we and what should we think about going forward, just in a broad sense? And then we'll get into some more specific issues.
Okay, so I guess we'll work backwards a little bit
Mm-hmm.
from where we are, from where we started. But I do think it's important to comment on where we started.
Mm-hmm.
The pandemic was this invisible threat that hit us from behind. We knew nothing about this new pathogen. We had some knowledge of its lineage because we were able to determine that it was part of the so-called coronavirus family. And we were able to do that because we had the sequence fairly early on.
Mm-hmm.
And that sequence allowed us to determine its structure. So by studying the genetic sequence, we were able to understand what proteins, for the most part, were going to be coded by those genetic instructions. So we had that knowledge, but we really didn't know anything about how the virus spread, or how deadly it was, or how quickly it would spread, or how we could protect ourselves. So moving forward, fast forward,
Mm-hmm.
we now understand so much more about the virus. We know more about the genetic structure, we know more about the mutations, and we know more about how to develop clinical interventions. But really, our ongoing challenges involve both public health challenges and clinical intervention.
Right. Right.
And that's where we are now. We've made a lot of progress in understanding the science, and we can talk about that a little bit later if you want.
Yeah.
But really, we're still in a ethical in terms of our public health challenges, and that's one of the reasons why we say no one is safe until we are all safe, because of this is a global situation, and it doesn't serve us if we have all the tools in terms of clinical intervention and healthcare systems here in this country if we don't consider what our neighbors are doing across the globe.
Right. Right. Yeah. So I'd like to just, I think, go back and forth a little bit between some broader issues and specific things, and talk a little bit about the immunopathology of COVID. So I think that, in talking about our immunological system, which I know in our previous conversations you've likened it to, this is our body's armies, to defend itself against, invaders. And of course, part of that is also involves how the vaccines participate in that. So, but before we get into that, I want to ask you, so a lot of the arguments come up between people like who ask, why do we need, we have a vaccine, why do we need another vaccine, why do we need boosters? And some of that I think is interesting, and I've heard you explain this before, about some of it involves understanding the different kinds of genetics of different kinds of viruses, specifically what the RNA viruses versus DNA viruses. If you could just talk a little bit about that in terms of, a quick explainer of why, some develop, variants and need, different kinds of vaccines and boosters, and others do not.
Okay, so I'll try my best because
Yep.
it's super complicated
right.
topic.
Right.
So as I mentioned before, we do know that SARS-CoV-2, which is the name of the virus causing COVID-19, that's a disease. SARS-CoV-2 is a member of the coronavirus family. And other members are MERS and SARS-CoV-1, and they are deadlier than this SARS-CoV-2, but they're not of concern right now. So all these viruses are RNA viruses, and they are single-stranded positive sense RNA virus. What does that mean? That means that you have genetic material in the form of RNA, not DNA, and you have that genetic material entering the cell and immediately making proteins, which is what causes the function, the behavior of the virus.
Mm-hmm.
And usually the RNA viruses versus the DNA viruses mutate much faster. DNA viruses, for the most part, are more stable.
What are some examples of DNA viruses?
So some examples of DNA viruses are smallpox, chickenpox, herpes, human papilloma viruses.
Okay.
And some examples of RNA viruses are polio, mumps, measles. So in general, the RNA viruses mutate a lot faster. But what happens with coronavirus? Coronaviruses are very large for RNA viruses compared to other RNA viruses. And instead of mutating, or we thought, instead of mutating so quickly, they don't mutate that quickly because they have a repair mechanism built into their genome, their genetic instructions, which means if a mistake is made when copies of the RNA is made, it can be repaired. Mm-hmm. So we didn't expect coronaviruses to mutate that quickly, but that's not what's happening. What's happening is that the virus is mutating at least twice as fast as we originally thought, probably even more.
Wow.
And so far there have been 12,000 mutations. I had mentioned 2,000, but I got my numbers wrong. It's wow. 12,000, I double checked. Wow. 12,000 mutations since the original virus was described. And so that means that we are causing variants. That's where we see variants. Now, there are different types of mutations, right? You can have a mutation that is beneficial to the virus, allows greater infectivity or immune escape. It's beneficial to the host where, the virus doesn't get inside, or it doesn't matter. So most of the mutations are nonsense. They don't matter. But when you have that many mutations, when you're looking at 12,000 mutations, statistically, you're going to have some that are trouble. So that's what's happening. We're having trouble because we're allowing that virus to propagate in unvaccinated people, right? And allowing it to mutate, and then that means that it goes back to someone, even if they've had a vaccine and they get sick, but they're not going to get severely sick, and they are not going to end up in the hospital. We can talk about the analogy. Do I have time to talk about the analogy?
Yeah, let's talk about the analogy, and then if we need to take a break, we'll just hold your thought and keep going after the break. So let's talk about that. Yeah.
So we go now into the vaccine development. So the vaccines are developed to neutralize the ability of the spike protein, which is what sticks out on the coronavirus, the spike protein, it's binding to the human cell receptor, which is the ACE2 receptor. Now, so the antibodies will interfere. They'll neutralize, they'll bind to that spike protein so that that spike protein, which is attached to the virus, cannot enter the cell, right?
Right.
So when you have mutations, you have a lesser ability of the different army of soldiers of the vaccine, because when you create a vaccine, you have what's called a polyclonal response, which means you have different B cell clones. The B cells are the ones producing the antibodies, binding to different areas of that spike protein. Now, the first time you have a vaccine, your immunization, let's say you have 10 soldiers. When you get your second dose, now you have 100 soldiers, which usually is log-fold increases. When you have your booster, now you have 1,000 soldiers. So the booster, even though it doesn't see the parts that were mutated, you still have some regimens that will see the original spike protein and neutralize it. I hope that makes sense.
Yes. So yeah.
I use a lot of hand gestures.
Well, yes, but I think that's a fairly direct, explanation, and I think helpful. And I think that yes, you can talk a lot more about the specifics, but I think we're gonna shift now.
Totally.
Yes, we're going to shift. So we're going to actually, Francis, we're going to take a quick break in a few seconds. But there's be much more to talk about when we come back from the break. Talk about, there are many issues with respect to, who's at greater risk and why. So when we come back from the break, we'll get into all that. So folks, don't go away. There's much more to come with Dr. Francis Santiago-Schwarz talking about the coronavirus pandemic and how we can move forward from here. So don't go away. We'll be right back. Welcome back, folks. We're talking today with Dr. Francis Santiago-Schwarz, a distinguished immunologist, talking about the state of COVID and providing us with some solid, pragmatic information and advice at a time when we're often confused because there seems to be changing information week to week from source to source. So thanks, Francis. Before the break, we were talking about, vaccines and a little bit about how they work with our immune system. Now I know that obviously the vaccines are not the same effectiveness with everybody. Who is and there are certain populations that are more at risk, and why is that? I mean, older people certainly are one of the more vulnerable populations, and then I know that there is some association with COVID and the increased risk of diabetes, and also with folks who are, in the obesity category. So talk a bit about the different, risks to different populations and why is that?
So early on in the pandemic, we realized that people who are elderly, or actually not even that old, greater than 50 years of age, were more prone to develop severe disease. And that's one of the reasons is because the immune system does not function as well as you age. So we are still unraveling a lot of those secrets, but we do know that there are decreases in the distribution of certain immune cell types that affect the immune response, and that actually even suppress the immune response in some instances. So then we realized also early on that smoking was a risk factor.
Mhm.
And some of the science behind that was related to the fact that smoking increases the number of ACE2 receptors on the cells in the airway. Remember, the ACE2 receptor is a human receptor that the virus utilizes, a spike protein utilizes to bind to a human cell and
Right.
get it to nucleic material inside the cell. So that was some insight into why smoking was a risk factor. Then there was another insight into why obese people might be more at risk for severe disease, and that was related to the virus being located in certain cells within adipose tissue, fat tissue. And so scientists found actual virus reservoirs in that fat tissue, so that it would sort of like sequester itself there and then and keep on propagating. And then the other thing that we actually realized very recently is that there might be a complication of diabetes post COVID, the long haul, the long COVID so-called scenario,
Mhm.
and that there's evidence that that's related to the virus actually infecting cells in the pancreas, the
I see.
islet cells, and actually killing them. And that's at least one mechanism. So we're learning a lot. See, I can go on.
Right.
So there's also the issue of autoimmunity and why
Mhm.
some people who are immunocompromised with autoimmune diseases may be at risk. And that may be related to different reasons, but one of them is related to the medication that they may be on that will compromise their ability to mount a good B cell response and produce antibodies.
Right. Right. Yeah. So we're learning a lot. Our previous conversation, you mentioned you have 1,000 articles in your library about this,
Oh
and
gosh.
yeah, well, it's as you've said before, it's a complicated disease. We know very little when it started, and we know a lot now. So we are learning, and we're learning every day. So one of the things
And we just need to be, I'm sorry,
yeah, no, go ahead.
but as a scientist, I am so humbled by what we don't know and what this virus and this pandemic is teaching us
Right,
in terms of what it is that we need to focus on and learn.
right, right, right. Now, in terms of the vaccine's effectiveness, obviously it varies from person to person. It's not as effective on some as others. We don't react the same to it. I felt that I've been very fortunate. I haven't had any reaction to either of the first two shots or the booster. Um, but I don't think that, from what I understand, that hasn't been the norm for a lot of people. But when we talk about the effectiveness of a vaccine, how do people, scientists, determine that, or those doing these studies? I mean, I assumed it was just sort of checking two populations. One is vaccinated and one perhaps receives a placebo, and looking at the incidence rate of infection. But is that the way they determine the effectiveness of it?
Well, there are three terms that are being put out there,
Mm-hmm.
and they can be very confusing.
Mm-hmm.
Um, and so there's vaccine efficacy,
Okay.
which measures a vaccine's capacity to succeed in ideal conditions, like the
Oh,
controlled trials. So
okay.
when you're talking about efficacy of the Pfizer or the Moderna, and they say it was 95% or whatever, its efficacy rate, that is a formula that was used that compared the experimental arm, the ones that were actually getting the vaccine,
Right.
to a control. So they have a formula, and it's strictly related to that. Then there is the term effectiveness that describes how well a vaccine performs in the real uncontrolled world when it's given to you or me or whomever. And then and so that there are a lot of variables involved, right? Because we have immunocompromised people, we have smokers, we have variables that perhaps weren't included in the control study. We have children now. We have a lot of different issues. We have situations where we're giving it to asthmatic people, or we're giving it to people who are not as nourished, or people who are in polluted areas. All these things. All these things in the real world matter. So the effectiveness is that. And then some people actually take that a step further, and they look at efficiency, which looks further at the measuring outcome in relation to the amount of resources used. So how do the resources available in a particular population affect the outcome of the vaccine? So all these things.
Yeah.
So age and all these things.
Right. Now, I know that there are still people who feel very strongly about not taking the vaccine, but I think it has been shown to still be the most effective, intervention. But and of course now there's a lot of discussion about the masks. So that there are people in New York, for example, and other places they're changing mandates about masks. What are some of your observations about the effectiveness of various protocols, from vaccine to masks to you mentioned things to me like hand washing, just simple protocols of protecting yourself and distancing?
Right. When this whole thing started, I realized that there were 3 Ds: dose, duration, and distance. And a lot of people were talking about that. If you're getting heavy doses of the virus, you're more prone to get infected. If you are 15 minutes or longer, you're more prone. And if you're close, you're more prone. Now with the variants, that's changed because these variants are so much more capable of infecting a cell that the dose is a lot less than it used to be with the original strain. So it used to be, so the spike protein is very interesting in that it has 2 domains. It has the what we call the head, and it looks like a tulip. And when it's closed, when it's not activated, it actually has to go through a conformational change, shape shifting, if you would say, to open up that tulip. Okay, and once that tulip is open, then it will bind the ACE2 receptor. Now, the original SARS, when the virus was produced, a lot of it was closed, and there wasn't enough enzyme around, because there are cofactors that are enzymes to help open it so that it can bind. But now with the mutants, we have a lot greater number of those virus particles open the tulip, so you don't need as many to get inside the cell.
That's why it's more infectious.
That's one of the reasons why it's more infectious. And so where was I going with this?
Well,
got lost.
Yeah, we were talking about what protocols are effective in countering the virus. So
okay, so once it gets inside the cell, then the virus takes over. Right? There are 2 different ways of getting inside the cell that we know of. One is, again, so that tulip is open, bound to the receptor on the human cell, right?
Mm-hmm.
And then the head comes off, and then the stalk of the spike protein now fuses with the membrane, the cell host membrane,
Mm-hmm.
lets that virus genetic material in.
Okay.
And then the virus starts reproducing itself. It takes over the cell's machinery and it reproduces itself. So vaccines are meant to neutralize step one, right?
Right.
But we also have ammunition that can prevent what's going on inside the cell. And I can talk about that if you want now or later.
Sure. Yeah, let's talk about that. Since we're there, let's go into it. Yeah.
Sometimes okay. So once that virus is inside the cell, so it's RNA, that's the genetic instructions,
Mm-hmm.
immediately gets translated into proteins, which is what is related to the biological function of the virus. So that virus is so smart that it makes this very long protein, polypeptide, but it also has encoded an enzyme that will help cut it into discrete functional proteins.
Right.
That step, I'll call step number 2, is what is now being targeted by a new drug.
Okay.
Developed by Pfizer. Oh no, was it Pfizer? Yes, it was Pfizer. And that's great because it seems to be working. Now, if we didn't have that drug, there are other drugs that are being developed that are downstream of that event, where you have the virus RNA being copied. Because every time you make a new virus particle, a virion, it has this RNA genetic material. So you have to copy it. One virus can yield up to 1,000 new virus particles inside.
Wow.
This. So you copy it, and that is called an RNA-dependent RNA polymerase, that enzyme. So the other drugs that are being developed target that downstream event. So we have ammunition. We have the vaccines, we have the antiviral therapies.
Right.
But we have really good ammunition, which is wearing a mask and washing your hands.
Right. Talk about that a little bit more, because I think that people say, oh, yeah, but actually it's very effective. So talk a bit first about washing your hands.
So one of the things that we know from before, from the coronaviruses and its structure, is that the genetic material is surrounded by another protein called the N protein, nucleocapsid protein, that protects the RNA, tucked inside like a fat bubble. And that's a membrane. And that membrane was actually stolen from a membrane inside the cell. So what happens with lipid membranes? They dissolve with soap. I mean, it's like one really good thing about this virus is that washing your hands with plain soap and water, the 30-second thing, will just, I have visions in my head of the membrane falling apart, and then that nucleocapsid protein that's surrounding the RNA dissociating, releasing that RNA, and then that whole virus particle falls apart. When the RNA is released, there are these enzymes all over the world, the outside world, called RNases that destroy the RNA. So we really need to wash our hands when we think we've touched something that's infectious.
Right.
Or,
Right. So when we see these scenes, in life or on TV of surgeons and doctors scrubbing, that's why, because it's effective. It works.
It works. It definitely works.
Right.
And I think that when you understand something like that, I think you feel empowered because you understand, this is why I need to wash my hands.
Right.
And I think that people are more compliant when they understand why they need to do something.
Right, right. So we're coming up for another break. So I think we're going to hold up, but I think when we come back, I'd like to talk about the other issues, which is, that people talk about a lot, which is masking and how is that's effective, whether different masks make it different, and also just the issue of social distancing, and how that plays into the continued
My pleasure.
pressure. So, folks, don't go away. We have one more terrific segment coming up with Dr. Francis Santiago-Schwarz. Don't go away. Much more to come. Welcome back, folks. We're talking today with Dr. Francis Santiago-Schwarz, a longtime immunologist who's talking to us about what we need to do to manage and move forward through this pandemic. So before the break, Francis, we were talking about various protocols and interventions. We talked about vaccines, and then we talked about washing hands. Let's talk a little bit about wearing masks and other kinds of protections. How effective is that in terms of, and what should we be doing going forward?
So we already talked about washing the hands, which
Right.
is critical, especially when you're at high risk.
Right.
So what masks do, there are different types of masks, and I know there was a lot of confusion about the mask. And part of that confusion was related to the fact that early on we didn't understand how the virus was transmitted. And as more and more data accumulated, we realized that the virus was transmitted via people who were asymptomatic as well as symptomatic. So that was something that was a pretty bad case scenario. But we also realized that in addition to droplets, see, it's a respiratory virus, so we had a clue that it would be via droplets like the flu. But the virus also is transmitted via aerosolized mist. So if I'm talking right now and somebody was sitting next to me and I were infected, even though I'm not sneezing or coughing, they would be at risk. I would be infectious. And so we realized that certain masks that were cloth masks that were homemade were not going to be sufficient because they wouldn't be able to trap, especially the aerosolized particles. So if you're masked and I'm masked, that increases the chance of protection on both ends. The way the N95 masks work is that they have different layers, and those different layers have different, allow different size particles to go through them, but they also trap via charge interactions the viruses. Now remember, the virus is tiny, but the virus is not going to be, if it were, it doesn't travel in isolation as a tiny virus. It travels encased in mucus or some sort of protein of the respiratory secretion, and also in an aqueous setting. So those particles are much larger than the tiny micron size virus particle, and they will be trapped effectively via those N95. One of the issues that people are talking about now is the quality of the mask, and even surgical masks are a problem because they don't conform tightly to the face, and the virus, especially if it's aerosolized, can come in through the sides and infect that way. Another thing that people don't realize is that you have receptors for the virus in the eyes, and you can get infected via the eyes as well. Now that's a tough situation to deal with, on a mass level. So what's recommended is that healthcare workers or people who are working in situations where they are caring for, let's say, an infected person or other high-risk situations should wear eye protection. I wear eye protection when I fly. We're not, on the air.
Right. Yeah. Well, I see many situations now, of course, especially with a lot of big sports venues where, I see people are not wearing masks. I mean, and
Mm,
that's just the reality. I think some cases they don't believe in them. Sometimes they're tired. I think sometimes they're just basically, assuming there's herd immunity for whatever that's worth. Um, but and I think it's also worth mentioning too that this is it's a continuing investigation, and so I'm glad you mentioned that there are other drugs in the system. Not just vaccines. These drugs that are designed to, intervene at different life cycles of the virus. I think these are important developments, and people should realize that we're moving forward. It may not be reported in the newspapers every day, but there's a lot of stuff that's coming out day by day. Um, and so I wanted to spend a little bit of time talking about, because one of the things that's been important to you that you've mentioned is just your involvement in educating people and educating, creating programs and partnerships and alliances to just bring information out there, just like I feel we're doing today. This is what we know. This is what we don't know. This is what's important. This is why it's important. I think it is one of the things you said earlier on, I thought is critical is people are more likely to, you know, uh, make changes that may be inconvenient or annoying if they know why. If they know what the why. So talk a little bit about and tell me about some of your programs. And you mentioned one program to me previously called the Ambassador Program. Perhaps you can talk about that, but other programs as well, and why it's important in terms of your overall philosophy about promoting health equity.
right. So my journeys have taken me into places where I value and I'm involved with community engagement to address health disparities. And education is a large part of that.
Hmm.
So one of the things that we did early on with the pandemic is that we developed what we called ambassadors program, where we had pipeline students who were part of the medical school, and this was all, of course, through Zoom. So we would give them some ABCs of the virus and COVID, what we know, what we don't know, why it's important format. And we would follow that educational activity with a town hall where the students then asked a bunch of questions. It didn't matter, just ask whatever you want to, just ask any question, it's okay. And we, at the end of the program, we made them ambassadors, COVID ambassadors. And some of them were undergraduate students and some of them were high school students. And they would go back, and it really touched us
Hmm.
deeply because they would go back and they would establish clubs. Even one kid at the university, like an ambassador club. So he would lead it and he would, get
Hmm.
back to us and ask us questions, and he would then empower in his mind other people, other students, like he felt empowered. And he would say, I understand so much now. And of course, yes, it's the tip of the iceberg, right? This is a monster, this is a bear, but every little bit helps. Um, One of the things that I have to say is that globally, um, the world did come together to make vaccines work,
Hmm.
because it wasn't just the scientist coming. So everybody thinks it was rush, rush. It wasn't rush, rush. The RNA technology was being developed for many years, 10, 12 years. And the lipid technology, which is the fat that the vaccine is covering the genetic material for the spike protein, um, that was independently being studied. So those two camps got together and they worked this out. So then they worked together, scientists worked together with governments here in the United States, all over the world. And not just the scientists and the pharmaceutical companies and the government, but also those volunteers. There are thousands upon thousands of volunteers who participated in these clinical trials. Without them, we wouldn't have had a vaccine. So there is a lot to be thankful for in terms of collaborative spirit, and I hope that we continue that type of collaboration that we see the fruits of the labor, because vaccines are right now our biggest hope. We have to stop this virus in its tracks. Well, that's too late for that, but we have to at this point downstream, we have to stop it from mutating further, because as long as it mutates, that mutation can represent an advantage for that virus, and our immune system is going to have a really hard time catching up to that advantage.
Right. So I guess there are thousands of mutations. So it's not clear what's beyond Omicron at this point. People seem to feel, rightly or wrongly, I don't know whether they're just tired, but they're feeling like, well, we're just going to live with this. But it seems that it still is important to continue with vaccinations and these other kinds of drugs. I guess there are some other developments with different kinds of pills. I don't know. I don't know enough about them to know. Are these, sort of, do they work the same way as vaccines, or are these other kinds of interventions, as you mentioned before, that intervene at different points in the life cycle of the virus? Is that correct, or is that?
Yeah, no, you're correct. That's exactly right. So the other drugs that are being developed are independent of vaccine strategies. It's those are preventative. These are actual interventions. So they're once a person is infected and the virus is inside the cell, it takes over the machinery. It goes through a life cycle. So those drugs interrupt that life cycle at discrete stages.
Right.
And there are also worries concerning that, because those drugs aren't totally safe, right? Because all drugs have side effects. We know that.
Right.
But the vaccines are now safe. There's so many people who have received how many billions of people have received the vaccine. And yes, there are issues, but they're very rare. And the benefits far outweigh the issues that have been reported. So as long as that's the best tool we have now. The mass, and we're tired of that, right? We're tired of that. And I sometimes forget. I go, I walk out the car and into the supermarket and go, where's my mask?
Right.
So we're tired. But
Right.
We need to get vaccinated.
Right.
As an immunologist.
Right. Well, we're coming to the end of our show. Unfortunately, we could have a lot more to talk about. Perhaps I just wanted to actually just mention, just in passing, that you've told me that, and if you look at my website, rowellresources.com, you could see some of Frances's poetry. So some of this is something that as she's been involved with, partly through the vaccine, just as part of your life, and talk about how do you manage life and what things do you do to get through. And so we don't have a lot of time to talk about that, but I thought you could see some of it, her poetry, on my Rowell Resources site. Click on 45 Forward. And I just want to tell folks, first I want to thank you so much, Frances, for a really quick study, a crash course in COVID, which I thought was still, very valuable, giving us some really helpful information about how to look at the disease and how to understand it without getting freaked out about it. So folks, if you missed my conversation with Frances, you can listen to it as a podcast at voiceamerica.com. Just search for my show 45 Forward and you can find it on Apple and Google Podcasts, Spotify, iHeartRadio. Go to my website, rowellresources.com. If you want to send me comments, if people want to get in touch with you, Frances, is there a contact that you're all right with, or?
Well, can they go through you?
Yeah, you can go through me, and I'll get in touch with her if you have any questions for her.
I'd be happy to.
Great. Okay, terrific. Okay. So folks, be sure to join me next Monday at 12 noon Pacific time and 3 p.m. Eastern time, when I'll be talking with Keith Allibone, who's CEO of Goods, Vintage and Antiques. He's going to give us an inside look at the antique business, and also think about how to declutter our homes as we get older, of stuff we've accumulated, and perhaps discover some unexpected treasures. So until then, folks, keep moving forward, 45 Forward.
Thank you for having me.
Pleasure having you.